8 hours, baby.
8 hours.
Hey, ding! Welcome to the Operation Grow channel. What week we in?
We are on week 7.
Week 4, right? 5.
Week 4 was growth. It's show number 7, so we've been going for a while now.
Right on. Yeah, we have been going for a few weeks now. I guess I'm counting plant weeks.
Yes. So, how's everybody doing this evening? Or to this afternoon, this morning, depending on where you are. It's evening here in the UK, so to all the UK followers, good evening.
What's up?
Who should we start off with this evening? Let's start with, well, if we've got a guest joining us this evening, which is Bear Grown, for when we dive in and talk a little bit more about, on the genetic side a little bit later on, hopefully. Get another perspective on the breeding side, which hopefully be quite good. So yeah, Bear, do you want to just give a quick introduction? Because I don't think anybody, well, people on here know who you are, but people watching possibly not.
Yeah. I'm Bear Grown. I'm a breeder with Land and Air Genetics. We're a fairly small company, focused mainly in landrace and some of the heirloom cultivars that have been passed around for the last 20, 30 years. Our main key or goal is preservation of the genetics and also creating some new true modern hybrids with some of the land races that we collect.
Excellent, cool. So that'll be quite relevant for the bit about finding stable lines when it comes to doing the crosses to get what you're looking for. I found some really interesting, some nice websites that actually do some good utilities for mapping out gene sequences, etc., in terms of genotype and phenotype expression. Punnett squares, which was quite cool. So moving on from there, Dr. Wriggler, how are you doing? So how's your week been? I've seen some quite good activity on the Instagram on the grow front there.
I'm doing pretty good. My week's been good. It's not rained the last 3 days here in Michigan. That's a plus. I mean, it's a tsunami here lately, basically. So the last 3 days we haven't had rain, which is awesome. But you know, as a farmer, that's something I've never asked for, was not to have rain, and it's pretty hardcore that like we're begging for no rain. And but yeah, so we got the Operation Grow. We moved them up into some different pots. Got a little slight leaf burn on some of them from sticking them under the thowies as well. It's always part of the fun, trying to get the hustle going. But yeah, we're just wiggling it out over here.
No, cool. I hope a couple of weeks back, actually, there was somebody in the chat channel that was asking questions. They had a couple of questions on the worm front. Hopefully they join back in on this one and see you on, because we did say they should wait for you and pick your brain, being experts on
worm questions, hit the DMs. Like, I will go for hours and talk to you about worms. It's way easier than trying to do it in the YouTube chats and this and that. Like, I'll send you pictures back and forth about worms. I'll try to get the temperatures and humidities right. And I geek out on worms like crazy. So
you got any worms in a bikini?
We can make that happen.
That's a whole news specialist website. So, Mad, how are you doing? I'm just going it down in the sequence it appears on my side, so hopefully it's...
Oh, I'm freaking tired and delirious. I've not had enough sleep in the past few days, like 2 days, yeah. Been fucking trimming my harvest, and it's a lot of work.
That it is. It's a lot of work to trim. It can get tedious, time consuming.
I fucking hate harvest. I like talking to them and feeding them. I fucking hate harvest.
Yeah, it's the one thing is, so I'm told that everybody enjoys only doing once. And the second time, they're not so keen. So I'm told. So, Mystic, yourself, we're going to have to jump back to you for a little bit more in-depth on the whole tissue culture side of things. But just a quick update from you on your grow. How's it going?
It's going pretty good. It's going pretty good. The girls are doing great. I got a bit of a thrip issue, though, and I had to run out and grab Mammoth Biocontrol. We're going to be spraying them with that.
That'll be a good test to see.
Exactly.
Have to tag them and see how it goes.
Yeah, so we're gonna see how it works. Where I'm living at, I deal with thrips just like fungus gnats. I don't deal with gnats, but I have the thrips, and I hate it.
Not
How
the best
much is
ones
that
to get.
bottle? I'm curious what their pricing is on that because they just released it.
It's $50, and this is a 500 milliliter bottle.
That's a lot more reasonable than the Mammoth P.
Yeah, and it's supposed to get rid of insects, mites, thrips, aphids. It's supposed to work on every different little pest that I would have to deal with here.
Can you tell us what the dilution rate is on it? Like how much per gallon in that?
Oh, shit. Babe, what does it say? Hold on, I can't read it. I don't have glasses. What? Oh, yeah. Also want to talk about, we just topped the ladies. We have the 6 out of the 10 seeds were females because we did the DNA testing. We're a little ahead of the game and we know exactly what we got. So we got the 6 females topped. They, number one was topped at the 5th node, but it's got very little branching. Number 2 has no branching at all. It's pretty sad, but it's the tallest one, so it's still stretching. It's not like branching yet. Number 3 was topped at the 4th node. It looks pretty nice. It's got a little bit of branching. 4 was topped at the 5th node. Nice branching. Number 5 is the best looking one out of the group there. It was topped at the 5th node, and that's my choice out of the group. And number 6 would be my second choice, and it was topped at its 5th node as well. So they're looking pretty good. I didn't measure them again. I guess I'm not trying to make mine bigger than yours or whatever. I don't know. I just haven't been worrying about the measuring. I keep forgetting on that part. I should have measured them before topping them, but.
But if you go and measure them now, I mean, it doesn't matter necessarily, so long as you just do it occasionally and made a note of when you topped it, that'd be good.
All right. And what was the dilution on this?
We'll still be able to plot it out over time.
It'll make 5 gallons, that whole bottle.
This whole bottle here will make 5 gallons.
It's not
So
much.
does that answer your dilution?
That doesn't seem right.
Yeah, so that's like $10 a gallon, basically. You got to fill that little top thing for 1 gallon. Is that what that comes down to?
I think so.
It's 3 ounces per gallon.
It's 3 ounces per gallon.
Oh, 3 ounces per gallon. Sweet.
Yeah, and there's 16.9 ounces in it.
Ah.
Right. So, Northern, how are you doing, sir? How's it going on your side?
Hey guys, can you hear me? Hello,
Yep,
can you guys hear me?
Good.
All right, cool. Yeah, no, things are going pretty good on our end. We're still sitting at 11 out of 10. Our plants are now in 1 gallon pots. We're thinking it's time to move them up to maybe a 3 or a 5. We topped all of them to the 5th node, except the 2 mutants that we got, or the double-headed ones. We're just letting those play out for a bit to see how we're going to handle those. And we've been battling humidity here. We've got a lot of rain, So my buddy Mike, who's kind of doing the bulk of the environmental controls, he's pretty much working at that 24/7 to try to get it a little bit more in line to optimal. We didn't really know, I guess, where our VPD sat up until we got this monitor. So now he's kind of going a little bit overboard with the details on it, but hopefully it'll pay off to get some good quality and a nice yield. They all look very beautiful and everything's going swimmingly. No issues with pests yet, and no fungus or mold. Hopefully with the high humidity it stays that way. But yeah, we're just having a blast and everything's looking really good on our end.
Excellent, it's good to hear. Yeah, that's the trick with suddenly all that data, as you say, racing around and trying to dial it in perfectly.
Yeah, I know he's had some hiccups in his last couple of grows, so I think he's a little bit paranoid about things not going well this time around. So I think for him, he's the type of guy. The more data he has, it's a good thing, but also it could be a bad thing too for some people. But
the OCD crew, it certainly becomes a bit more like a crack habit. But at least you can set it so that you can get the alerts before, you know, if you set that parameter and you can go and make sure things are okay before they get really out of control.
Yeah, I know. I think he said he hit 99% last night, but he was also testing out a new batch of RSO, so he kind of might have overdone it a little bit on that end too.
Yeah, no, the trick there, if it says that it's hitting 99% humidity, that can sometimes indicate that moisture is condensed on the sensor, and it's something for any type of humidity sensor to be aware of. The trick then is to go and have a look at the charts and check to see if the ambient temperature has crossed the dew point line at any point. Because if it's crossed through that, and then you've had condensation form on the sensor itself. And we may need to just get you a replacement sensor sent out. It's just something to be aware of. If it only did it briefly, I mean, it can happen. I mean, it happens here. I get it happening in the greenhouse quite often when it's been raining and that it will hit 99% humidity.
Yeah, well, I think we're sitting around the 70s for the most part. He's trying to dial it in a little bit lower than that, but he does know that there is a bit of spike, especially when the lights come on first thing in the morning. There seems to be a spike of humidity.
Yeah, that's as the plants are waking up and all the stomata open and they start transpiring at that initial kick. So it's not, as you point out, it's not just as the lights go off and the temperature drops and you get that humidity spike from the drop in temperature. As the lights come on and all the plants get going, you get that humidity spike as well.
Yeah, but the plants look really good and they're growing quite a bit. We're hoping to get them sexed before we transplant them out for obvious reasons, but in terms of that, well, we're probably going to be relying on some of the more experienced growers here to see if they can kind of show us, I guess, male from female as early as possible before we transplant them out. But yeah, we're looking forward to it.
I always just ask my magic 8-ball, and it's fucking right on every time.
Yeah, so that's about how we're doing right now.
Great. Oh, it sounds like everybody's coming along well. So, Tanasi, I see you're busy measuring away there.
Right. I didn't know how humid my room got at night until I got the minders in there. And I was able to see when I go to bed and stuff, lights are out. It got fucking like 80s. Some of them got 90s in the tent that I was running. Didn't even have a fucking clue.
Oh yeah, humidity goes up crazy at night.
Yeah. Took a minder though to tell me.
Yeah, and then the thing is, when you just pop in and have a look, and you'll see it says 80 on the one on the wall, as that was the max. And you know, I think most growers in the back of their mind go, well, they probably only just got there briefly for a split second, not realizing it sat there all night long, and it got there really quickly. So how you doing there on the growth front?
These babies are looking fucking beautiful. freaking green. They're short and tight, nice nodal spacing, and they all seem to be symmetrical. As opposed, I just had some other testers from him, like the Sergeant Slaughter and the Dirty Gym Bag. Those were kind of like how Mystic just described hers. There were some that have good branching, some with none, some with unsymmetrical branching. All of these here, man, seem to be like symmetrical and fucking beautiful.
Didn't you have a mutant that like self-topped itself?
Say what?
Didn't you have a plant that like self-topped itself, you said? Or
Right. I have one of them over here that double sprouted out as if I topped it. Number 2 here.
yeah, because I got a weird little mutant that's can kind of like split itself at the top too, and it's got a weird little middle leaf. I don't know. I might run down and grab it.
I usually do this, like if I had a clone or something, I would do that to it. Let's see.
Yeah.
I can tell.
Yeah, I had one that fucking grew up like that too.
But that's what you want, I feel like. 4 main branches like that.
Yeah.
I still got the little 2 dude over here, but you can tell that dude is not the biggest. He's definitely, this guy's taking over. I'm trying to bring him out the way, though. Give him some light.
I was doing that same thing with mine. I was like, get out of the way. Give him some room.
Yeah. Give him some light, you fucking savage. You're killing your brother. And it's
But it does sound like the rest of the ones you've got there, they're all looking kind of the same, so sort of similar height, similar dimensions. I mean, if you looked at them, you would say they're all looking pretty much the same.
4 is 5 inches tall. Nobody's over 6 inches.
I'll be right back. I'm going to go grab mine.
Yeah.
Excellent. So, Richard, how about yourself, sir?
How's it going?
Yeah, going good. How's your grow going at the moment?
It's going good. My 6 are still growing real nice. They're all really short though, like Tanazi was saying, all under 6 inches, really tight internodal spacing. I do got one weird one. I don't really know how to describe it. I'll have to throw a picture up later of it. It's kind of split at the top, like Monkey was saying, and putting off like a 3rd extra leaf. It's really weird. I have them all under CMH lights though, and different Kelvin bulbs. So I don't know if that's keeping them really tight or what, but they're definitely no more than like a 1/2 inch at the most internodal spacing.
Right. Same here on mine.
I do have one.
Yeah,
I have one that has a weird split leaf as well.
do you?
Yeah, it's just one that has one weird. It's like an extra, like a split in the middle of it.
Yeah, I don't even know how to describe mine yet. It's still kind of trying to figure itself out, so I can't really describe it yet. But I'll throw a picture up for sure. It's pretty cool. But yeah, they all look good.
What's the cross of the Jamaican Bobsled?
Jamaican Hash Plant times Space Dude. Am I correct on that? Space Queen or Space Dude? Let me double check.
Space Dude is just the male of Space Queen.
Yeah, that is space.
Same.
Proper.
Yeah. So, and then out of your ones that you're seeing there,
Space
Richard, I mean,
Dude.
again,
It's Space Dude.
sorry.
Go ahead, Peter. I interrupted you.
No, that's cool. So, Richard, on your ones as well, if you're looking at them, are you seeing like any that stand out, or are they all visually looking kind of similar?
Oh, they all look pretty similar. They're all super short, stout, bushy. Some are a little wider than the others, but no, they all look very similar.
Cool.
I'll put some pictures up today of them.
I was expecting these to get super tall, but they're not as
Yeah,
tall.
no, mine are nice and short like that. Yeah.
And yourself, Stoney? How are things going there, sir? I see those AirPods definitely have done a nice job on the roots. That's for sure.
Yeah, things have been going pretty good. This is some of the root development that I get out of those pots. And I had a question about whether or not you have to break all this up just to be able to transplant. And here's it shows you. I've lost very little roots here. Most of these were just teased apart, and they actually hang down a good little bit. But then I'll just gently set those in to my. I was putting these in 1 gallon radical bags, so I just potted mine up. I think it was 2 days ago. So yeah, good growth. They are staying short and squat. The branching has been pretty awesome. So yeah, it's been a good time. There's a good shot of what the branching looks like. So they're staying nice and squat and compact. But I don't know. I think we might get shocked and flower, and these things just take off. So we'll see.
Mind your head.
Yep, I was just going through here trying to figure out how to kill off my plants here because, you know, I've still got all 10 in my journal, but I figured it out. You have to go in through the room details. So yeah, I'm just trying to get my journal updated here as we're all chatting, but things have been going good. I got like I said, I got all of my 5.5 plants repotted. We're just going to call the mutant tulip lip plant like about a half a plant at this point. So I got 5.5 plants looking pretty good, and I gave them all a topping the day before I did the transplant. It wasn't the kind of thing I could weigh, though. I just knocked the nub off the top.
No, that's fine. When I've done it, I've just put it in as like 0.1 of a gram when I've topped my chili plants.
Yeah. So yeah, everything's been going pretty good. Liking what I'm learning about.
And again, anything else? I mean, there's obviously everybody. Well, not everybody, a few people.
Say what? You're breaking up.
Other than the odd runt one, how's how the rest looking? Are they all looking again similar height, similar structure?
You're kind of breaking up there. I don't know if anybody picked that up.
Yeah, no, he's breaking up. Yeah, other than the one runt, I would say I got three pretty average plants. They're about 9-10 inches tall, so 160, 170 centimeters, something like that. And then I've got 2 that are just really taking off. The roots on them were even a lot bigger than these plants. I really kicked myself. I didn't take a lot of pictures on this JB6, but it did have so much root development. I just had to sacrifice some of the roots to get it repotted. But as long as you're just peeling off some of those bottom roots, even though they look like they're just really nice roots, I don't even get any droopy leaves after a transplant. So doesn't seem to really mind it too much. But yeah, everything's been going good. My growth has been real good on those 5 plants. So. And yeah, I think having the minder unit to help me dial in my room has been a nice benefit too. So I'm learning a little bit more every day.
Oh, good to hear. Good to hear.
Man, those are pretty bushy. They look pretty nice.
Yeah, they're bushing out pretty good. I think there's just something to, having the roots air prune themselves. And, there's a lot of different things on the market. But the only one I'm not a fan of is the fabric pots because I typically have to end up cutting them off, I feel like. So I think they're a little wasteful. But other than that, when your roots air prune, then they grow back. You get 2 tips every time a tip dies. So you end up being able to feed your plants a lot faster just because they have more structure to be able to intake all of that. So if you've never tried it, you might just grab 1 or 2 and just, do a side by side.
And how do you find those for root aphids and other types of root pests?
I haven't had any problem with any root pests. The only problem I've had with roots recently was drowning them, but that was on me. I up potted into too big of a pot a little too early, and 4 times a day waterings was just too much. And then to make matters worse, I was at the bottom of the bin on the cocoa, and it gets to be really finely milled down there, especially if you went through a couple of bales. So I think just the combination of that cocoa that was really fine and could compact a lot tighter and really hold too much moisture just kind of spelled disaster. And I got them to all come back out of it, but that's the only root issue I've had. So I haven't had pests yet, and it could just be because they get, they're not buried in the same way that you would be buried in a pot. But, knock on wood, it's probably just I've been lucky. So don't bring any bad juju on me, Peter.
Yeah, hopefully not. Absolutely. I always just find the watering in those was always interesting. I mean, it was a bit of a challenge sometimes, unless you pack them properly. Watering them what was problematic, I found.
Well, I water from the bottom. I water from the bottom with the flood and drain. So yeah, I think if you're putting in water in the top, especially the radical bags are fine. The holes are really small. And as I buy more things, I've just been going to that. But yeah, the holes can be pretty big, especially in the oxy pots. They seem to be, or no, the air pots seem to have the biggest holes. I like the oxy pots, which should be easy for you to get your hands on because I actually have to have them shipped over from the United Kingdom for me. But I've got the my 3-gallon and 5-gallon pots are actually air pots. Now I don't know, one way or the other.
Ooh, that bell.
Is that too high in your headphones?
No. I'm still scarred, dude. Still scarred. Excellent. Underground, how are you doing, sir? How are things going in your neck of the woods?
Well, everything is going good. After the AC incident, 2 survived. They're kicking it. They got a T yesterday, so we'll see how they do.
Good.
And in the process of moving, so packing and doing a bunch of other crazy stuff.
Oh man, it doesn't help.
Nah, it really doesn't.
And yeah, especially when you're growing as well, moving is always a pain in the ass. It adds an extra dimension.
Yes, especially with the AC going out and moving stuff in and out, trying to make sure I can get things out without neighbors noticing, because I'm still in the red one right now. But the good part is I will be legal soon. Saturday to be exact.
Hey, even better party around yours then.
I got some crazy moving stories.
Right. Well, excellent. So good update. So they're bouncing back. The 2 of them just quickly out of interest. I mean, they're looking the same. I'm dead keen to, as I think it's become obvious, understanding what that phenotype variation across the board is to really get an idea as to how stable the parent lines were. Because everybody really should be seeing similar phenotype expression across the board. And by the sounds of it, the short, squat, fairly stocky, apart from you, Mystic, you seem to have some odd ones, but generally it seems to be kind of squat and stocky. It seems to be the general thing I'm picking up on.
I got one that's 15 inches tall, and it's 15 inches tall, and it's 12 inches wide from leaf tip to leaf tip. And that's my best looking, nice looking plant.
Something about that fucking West Coast air or something. Shit.
Yeah.
And that tube was 14 inches.
Illegal steroids.
Yeah.
Haha, we need to have her tested. She's fucking cheating.
My shortest one is 12 inches with a 10-inch leaf span. We're using the Sonic Bloom as well.
Well, that'd be interesting. I mean, now I need to run the same ones again in another tent without the Sonic Bloom and see what happens there. We get the true testing going. Now, on your week last week, Mystic, you went off and got more knowledge on the whole tissue culture side of things.
Yeah.
I have not been back. I've been back once. I need to make some time to go back over there. I've had a busy week. I went to a Revolution concert at the Avila Beach Golf Resort here on Thursday, and I'm an Uber driver, so it's been really crazy around here with summer. You know, I'm right here by Pismo Beach, so I've been super busy. I need to schedule some time with him to go back to actually learn to take the cuttings and to clean them and put them into the when I went over there, I learned to make some of the media, and we put it in the little containers, and we put it inside the what's that container called? Pressure cooker. Yeah, to sterilize everything. So when I go back over there, we'll be getting cuttings and putting them into the sterile environments.
I'm very interested in you finishing this so I can get more details from you, because it sounds a lot like integrating it with slightly some mycology skills, like agar type stuff and pressurizing to keep sterilized. And that type of stuff. So I
don't know nothing about the mycology side of things. So if I learn this, I guess I'll be learning a whole lot, huh?
Yeah. You do understand it a little bit better, maybe. I don't know. Easy.
What does seem interesting about the whole tissue culture bit is this is where you get to get rid of viral infections that have occurred in the plant.
Yeah.
Because one thing where plants differ from animals and us is once they get infected with a virus, that's it. They cannot defend it off. They just live with it. So variegated leaves, where you get the green and the white coloring in the leaves. Is it variegated? I can't remember if that's the correct term. That's due to a viral infection, or where you see some of those flowers that have multicolored petals. In some instances, what causes that, in tulips, what causes that is a viral infection. And it's now what they actually try to do is then pass that on because they want that happening in the flowers. But with a tissue culture, you could remove it's at that point where you can actually get rid of viruses in the plant. So I remember in the last time show we were talking, and there was mention of someone having, I want to say, scrubbed it clean, scrubbed the line clean, getting rid of some of the unwanted, but could possibly have been viral infection that had gotten into it over time. So 6 or 7 generations on, as far as the mother is concerned, if you're taking cuttings from that continuously and it does get infected at some point, then all the cuttings going forward will carry that infection. But doing it with the tissue culture, you can get it cleaned so that you don't have any of those traits passed on that are, as an example. That's one of the things I've been able to sort of figure out from looking around a bit.
Yeah, to me, it's basically kind of like resetting the genetics and regrowing genetics you can start from a very small piece and it just regenerates itself. It's just amazing what we can do.
Yeah, I watched them doing with an African violet. Well, it was in that video I posted, but doing it in an African violet, and it almost seemed like every cell that was in the sample that they took suddenly started growing into more little African violets. So you have these clusters of
chopping them off to make more
that form up that just then start to grow into more and more and more. So from one little sample, you can grow 100 plants all of a sudden.
Yeah,
How long do you have a viable? One question: how long before you have a viable plant that you can actually grow or actually take clone cuttings off of to start propagation with?
It depends on I think, the media, and you can keep it in. There's different stuff that you add to the media. There's different sugars, vitamins, food, rooting hormones. There's different things that you can. I learned that you can actually. I think you can. I could self them on potentially and take 8 months for it to be able for me to grow it back out. But then I could also take that same one and it could take a couple of weeks.
And the difference is you can get it to generate thousands of offspring. I mean, when you take the sample, you can create thousands of new little plants from one sample. So instead of having to go and take thousands of cuts so that you can propagate from cuttings, I mean, you take one cell, a small sample of the plant, and using tissue culture, you can suddenly generate
thousands
of them. Yeah.
Okay.
And it could take 8 weeks to get up to a point where you've got a plant that you can put in the ground, but you're also like, okay, I've got 3,000 plants in 8 weeks. It's a fairly quick turnaround.
In like 2 to 3 weeks, you can chop that piece up into like several other pieces, and therefore also let them also continue to divide into other pieces. So you could make so many clones off of one little tiny piece.
Yeah.
Now, what's the percentage of viable stock from that? Like how many actually turn into viable plants?
From what I've seen, when you do it right, as with everything, then you could have 100% success rate.
As long as everything is sterile and everything is right how it's supposed to be, you'll have your 100% success rate. It's just a matter of contamination and bacteria. If you get anything in there, if any contaminants, then it's not going to have success.
Yeah, and you can have odd mutations occurring because of the piece of the plant that starts to regenerate. I believe it's only the meristem that can that's the actual bit that can turn into any piece of the plant, and it's also the only bit going back. This is the bit about the viral infection piece. It's the only bit of the plant that would potentially not be carrying any sort of viral infection in it. So there's a very small piece that you can use, but from that it will generate quite a bit. So I don't know enough about it, but from what I've seen, you can, if some bits of the plant may not work as well as other parts of the plant for the process.
Yeah, I just heard you could use a leaf, a stem. You could use a piece of the root, different pieces of the plants. I don't know how well. I'm just learning about it, so I don't know how well which one's better than the other.
Yeah, see them say when they take it on a leaf, on all the videos I've seen of them taking it on a leaf, they've all made sure that they included a piece of the leaf vein in the sample that they were taking. There's possibly something in that tissue cell structure that you're after, for instance.
Yeah,
the paper that I read, I've seen when they just taken the leaf sample, they didn't have anything viable. Like what Richard was saying, what Peter was just saying there, when they take that, I guess the main part of that stem of the leaf, they have more viable product from it.
Sounds right.
Yeah. No, I think it's the way that they, you mass, and that's almost true cloning at that point. That's not taking cuts. That's true cloning.
Yeah, I understand that you need a part of the stem to be successful. Also, each cultivar is different. There's, you almost need to build a recipe specific for each cultivar. I've heard there's a lot more unsuccessful goes at it than success at this point in time. Even bringing in some of the best scientists that have been working in other aspects of agriculture doing this for years, they're kind of come to a point where they're almost stumped with cannabis as a broad base. There's been a lot of success with singular cultivars. Some haven't been able to have any success on. So there's a lot of honing and that needs to take place within the tissue culture realm. But the ones that do take to it, they can hone in on that recipe and they can replicate that success over and over again, but just with that specific cultivar. and going back on the you said variegation at one point was a viral infection, and that's how the plant regulates and deals internally with the calcium on those cultivars. Now there's a virus that looks a lot like that, and that's the tobacco mosaic virus, and it can give the impression of the variegation, but it's a lot different in nature.
Yeah, no, absolutely. I stand corrected in terms of variegation. Wasn't the correct term, and it wasn't referring to cannabis. There are some plants where like hostas is a good example, where they have like dark and they have dark leaves, but some of them will have light patches that run through them, and what's causing that light patching to run through those through them
usually they come through as a white more than a yellowing.
Yeah, and you get it in certain plants, and I just couldn't remember what the name of what it was. Yeah. And eventually, in some plants, eventually they manage to get rid of it, but most of the time it gets kept, and then they start specifically trying to breed for it. So in tulips, it's called the tulip breaking virus that gives it the multicolored flowers.
Speaking some knowledge I don't know nothing about.
Mr. Beargrown, I got a question for you as an avid breeder. Now, have you in your breeding processes noticed that different cultivars receive the pollen better on different days of your breeding cycle than others?
No,
Or
not necessarily
like a certain day?
on days. I've seen from one male cultivar not being accepted well by certain females, but the day of pollination, I mean, as long as there's a stamen ready to receive, there's no issue usually, as long as it's viable pollen. High humidity conditions, because water is the main enemy of pollen. If you're running high humidity on a certain day, maybe you could have a lot less of a success rate than if you were pollinating in a dry environment. But normally generally speaking, usually right around week 3 is right about when I want to start pollinating, and I usually pollinate over a succession of 3 to 4 days, sometimes 5, depending on how much pollen and how many plants I'm pollinating.
Okay, thank you.
That's pretty good information. I've never pollinated anything on purpose before.
It's so much fun to try it.
I recommend it for everybody, at some point to, keep a male, watch how it grows, and, pollinate something and grow those out. Not only will you be proud of what you're growing because it's yours, but it'll also give you a little bit of insight of what you'll see, because you'll go, okay, I'm seeing what came from dad, I'm seeing what came from mom, and it can expand your interest in the plant.
Can you keep a male plant like a female plant, like as a mother? You know what I mean?
Oh yeah, no, you definitely can.
So you just keep the lighting right, and it'll never spit out pollen, or?
Yeah, you keep it in. It's best to keep it in 24 hours of light. You can get some pre-flowers going, and males will drop pollen eventually. When I'm keeping them in veg for long periods of time, I usually pick any male flowers that start coming out. But
I see balls, I kill them. I'm like, oh my god!
Right.
So, a question for you, Bear. While you're doing your selection process, then, and for anybody else who's breeding, do you use things such as punnet squares as a way of better understanding what you're starting to cross and how to better, how to
I mean,
put together.
in a sense, it's kind of all ran through my head. I'm I guess what you would say more of an intuitive type breeder. I go off of what I'm visually seeing. Also, I know the backgrounds of my plants. And it depends on your outcome you're trying to go for too will determine on what you select to be taken to what. But at the end of the day with genetics, you're trying to stack a certain genotype. You're trying to stack a certain gene together so it expresses out in its progeny. So what you're looking for in your keeper female, say you're trying to increase, shorten the flower time on a certain female. Well, you're going to want to look for a male that has a lot of that same genotypical expression as the female. But you're looking for the male that will show the pre-flowers and flower out at a shorter period of time. Hoping that'll transfer over into the female.
Yeah, precisely. And just a quick question then for people on the show. I mean, do you know what a punnet square is? And just to jump, because I mean, that's a really useful, especially if you're starting out in plant breeding and you want a way of sort of trying to work out what the process should be, and Bear, step in and correct me at any point here.
Yeah, look, and
it's a very handy
Mendelian
tool.
Genetics.
It's
really it's basically you've got a blue bucket and a red bucket, and you cross those two colors together. You should have an expression of red, blue, red, blue, red, blue, red, blue, and how those come out are that would be the progeny. Now, a lot of the cultivars you're seeing now would be already red blues. And when you cross those, you get a multitude of genotype and phenotypical expressions. I'm speaking like polyhybrids.
Yeah, so for instance, I've got a little utility here that starts to show how this all fits together, and if everybody can see it. So using an example of, let's say big buds, and it has one plant has big buds and has big leaves. Now, if it was a pure stable line, both parents would have the same genotype, and if they were crossed, everything would come up the same. And it would be the same again if both parents had big buds, but let's say small leaves and big buds and small leaves. They would all come up 100% the same. And if you kept crossing them, it doesn't matter how many times you cross that line, everything comes up the same. And that's just using two genes in this case. And this is how the Punnett square does it. You take the alleles of the parents, and you create a grid that generates every combination from what's possible here. And in all of these, they will all express the same. At the end of the day, they all come out exactly the same. Doesn't matter how you mix them. But if the example you were using a minute ago, Bear, with a short flowering period.
Can you zoom in on that? I don't know if we can really see that.
Is it really small, is it?
Yeah. It's tiny.
Let me see if I can make it bigger.
I was gonna say I can see it pretty good, but my screen's 55 inches, like 3 feet away from me. 4K.
No, I don't think I have an option to zoom here.
No full screen?
No, unfortunately not. So this is not showing up too big, is it? Let me see if I can.
Well, I'm on my gaming computer, so I can see it. But yeah, no, a lot of people probably couldn't.
There's hotkeys that does it, right? Like a shift plus.
Yeah, I don't
Yeah,
know.
but this is inside an IDE editor that may not. Well, hang on. Actually, if I do this, does that make it bigger?
Oh,
Yeah,
yeah.
there you go.
Yep, love
All
it.
right. Okay. Cool. Okay. So let's say we have. Let's create two strains that are separate from each other, where there's features in each of the strains that we're interested in. So if we had one strain that was big buds but had a long flowering time, well, let's say the short flowering time was a recessive gene, so it had big buds but a long flowering cycle, and we had another plant. Hang on, I'm going to get this the wrong way around, aren't I? For the sake of this example. Okay, so it had big small buds and a short flowering time.
While you think about that,
And we have another one that produced big buds but had a long flowering time. Yep. And what we're after is we want to have something at the end of the day that creates big buds and has a short flowering cycle. In this example, it's going to come out in the very first run. The very first cross from that would give you every single strain that came out would have big buds and actually big buds in the long flowering cycle.
I think there's a song in there somewhere.
Yep.
There is. And I should really work on these examples a little bit better beforehand, because now when I'm trying to do them straight off the top of my head, it's a little bad choice.
It's fine. Here in America, we get killed by PowerPoint, so at least you didn't have a PowerPoint presentation ready.
See, I'd rather see your PowerPoint presentation.
Uh-oh, you just got downgraded to below PowerPoint. That's not good.
All right, an easier example is we've got a plant with big buds and big leaves, and another one with small buds and small leaves, and using the small leaf and the small buds as the recessive genes. And what we want to come up with is a plant that produces big buds and has small leaves at the end of the day. So on the very first cross that you do, and they're both pure, so it doesn't matter what I did with either of those two parents to start off with, and the way I validated that they were stable is I crossed them amongst themselves in their own population, and there was no variation in the offspring. They were all producing, both parent lines produced pure strains of, one was big buds and big leaves, and the other one was small buds and small leaves. The very first cross you do with them, you're going to wind up with big buds and big leaves on every single plant. They'll all have it, because the big bud and the big leaf bit is the dominant gene. So every time that appears in the cross, it takes dominance. So out of this entire cross, you'll actually, out of all of, you've got pretty much only one genotype and only one phenotype being expressed here. But out of all of these offspring, you'd only have one phenotype coming out. And that phenotype is big buds, big leaves. If you then take one of those, so now we take one of those parents, and the cross now is that each parent has one dominant big bud gene and one recessive, and they have one dominant big leaf gene and one recessive. And you'll see it starts to generate a few more variations, whereas previously it was 100%, they were all the same. Now this is your F2 generation that we've just created. And the F2 generation suddenly shows quite a spread in what we're going to get in the offspring. you're going to get one pure big bud, big leaf, and you're going to get one pure small bud, small leaf out of that cross. And then a whole mix of them in the middle. And what we're trying to figure out somewhere along the line is you're now going to have to pick, when you're doing the breeding, you're going to have to try and pick one that does big buds and small leaves. And we'll have one. I mean, there is one here that does big buds and small leaf. And if we were lucky and picked that one and ran it over and over, because the small leaf bit is recessive. So we know every time that we cross that one, we'll always get small leaves if we crossed it with itself. But the big bud piece is unknown because that's dominant. This one here would also give big buds and small leaves, but if you cross that one with itself, you wouldn't get a stable line. It would start to change all of a sudden. Do you follow what I'm saying? So if I take that one.
Exactly. That's why selection in the F2 generation is the most important. You really have to select your gene set properly, or your further generations will be kind of a moot point. You'll make a lot more work for yourself in the long run.
Because each one of the parents
possibly never hit your goal.
Yeah. I mean, that's just it. I mean, you've got to get lucky. I mean, you really do. Theoretically, you'd have to take every one of these and cross them with one of the parents so that you could work out which one of the genes was. I mean, I guess that's where the advantage of now back crossing it with one of the parents comes in. Because I know I can force the recessive line to come out quicker of one of the features that I'm after by back crossing it with one of the parents if I know it's the recessive feature.
Yeah, I mean, when you start doing back crosses, not only are you, stacking the genes for what you want, but you're also stacking the genes against yourself on what you don't want. All the recessives, all the nasty stuff will start stacking up and coming out too.
Yeah. And that's why I'm seeing people suddenly talking about, I've just seen on a few that we follow and whatnot, got F2 seeds of this. And to me, I'm thinking, hang on, that pack of F2 seeds is probably the one that's going to have the most wild variation in it. Your pack of F1 seeds should all be the same. Then the F2 ones will start to get a bit crazy.
Yeah, the F2 is where the true hunt is.
Yeah.
If you like a wide expression of plants and you like to hunt, F2s are what you're looking for.
But should you be paying, and then my question would be, should you pay more or less for a pack of F2s? The pack of F1s, true F1 hybrid, so they should all be the same.
Yes. Yeah, the F2s, should definitely, it's hard to say because, you still take your, you still have your selection process. There's still work. There's still time. To say that one generation isn't worth the same as the other, that's kind of a hard statement to make. Unless you're talking like something that's been worked for a long period of time, a lot of generations, then that I can see being deemed worth more than F1, F2, F3. But the F2s, it should be customer education. You should know going into an F2 seed stock that you're going to see a very wide genotype expression and, a wide phenotype expression within those genotypes. And, some people are pheno hunters. They are looking for that rare carrot. And, that's where you're going to find it more in the F2 generations than you're going to find it in the F1 or F3, F4 progressive generations. You won't be finding anything that rare coming up. It's the F2 that you'll find your pheno. I think they all have their value, and it's all what you're looking for as the customer should determine on what you're purchasing.
Yeah. And of course, I mean, just to iterate the point, the thing about that first little example there is we were only looking at 2 features just to show how, when you said that you blow it out and you go, well, I'm looking for a bit more than that. And let's say, using what we were using earlier, maybe we add that it's purple, short flowering, THCV, which is popular, let's say. I mean, it's enough that it's going to struggle to show it.
Yeah. No, and that's why
all of the variations that spin up, and it's like, wow.
And as a breeder, you shouldn't be going, you should be starting with a stable stock that you're going, okay, I want to make this one change, and you target it at that. You shouldn't be going, well, I want to make this cultivar bigger flowers, shorter flowering time, and I want it purple now. You're adding a lot of variables in what you're trying to change. The more you keep the same and just try to change one variable at a time, the more success you'll have.
Yeah, exactly. Questions from others, from online, from the chat? Yeah, I agree with AK420 there, Dave. With the polyhybrids, that's the thing. The F1s aren't genuine F1s anymore.
No, with the polyhybrids that you see today, they're basically, you're buying a, what I would coin an F2 generation. I mean, because there's just going to be so much genetic spread and expression that comes out in those polyhybrids. I mean, and that's why, the term pheno hunter is out there, because you really are, with these modern cultivars, you really have to search through to find something that's actually worth grabbing. The diamond in the rough.
Everybody looking for gold.
Exactly. And we're selling the equipment that helps you find it.
I love pheno hunting. I just wish I had more space to do more.
And as a breeder's point of view, these modern polyhybrids are a nightmare and basically virtually worth nothing to try and start a breeding program with.
Yeah, I had that one a few weeks back where I showed the what's the expression, the lineage, but it's got a different term, of some of the strains, and just showing you pick one quite far down, what looks down the line, called keyline pie, and you don't realize that that's got X, Y, and Z, and further up its parental strains have all got, they say, Durban poison in it, and then you have another strain called something completely different, which, unless it has, using Durban poison as the example, because so many strains today seem to have that in it, have that in the parents' line, well, they're not genetically diverse enough to be used in a if you cross them, they're not genetically diverse enough on that first cross to produce a true F1 hybrid.
I guess I didn't follow that fully.
The only way
you're saying 2 separate cultivars that parentally can be traced back to Durban, you're saying wouldn't have enough genetic diversity, like 2 F1 hybrids of Durban, are you talking, or?
Well, no, I mean, let's say you took, if I took a strain where I traced the lineage back and the root lineage from it, let's say it started off with Malawi, and I don't know, you have to say Malawi was one and Afghan Kush was another of the parents. So those are the 2 original parents, and throughout the entire line that generates the strain that I'm now working with, let's say, at no point was Durban poison used in any of those strains. and I can actually work. I can see everything that was used in it, and then the other strain that I want to cross with, I've made sure that there is nothing in that line that also appears in the other one's parental lineage.
Right.
So I have two completely genetically diverse lines that I'm bringing together.
Okay.
So I don't run the risk of flagging up a recessive gene.
Right. Right.
That's what I mean by having, and that creates a true F1.
Oh yeah, the divergence in the genes.
And the problem today is everybody is chucking.
They're all playing in the same gene pool.
Yeah, and then they're taking
West
something.
Virginia going on.
They don't know what it's called, or they give it a name. Bob's
Well,
back
everybody
end
likes the
snack.
same sandbox because they know what they're going to get. They like the tried and true. Nobody wants to go out. Everybody's scared to go out and try something new because they don't know what they're going to get. So, they stay in that safe little sandbox where, mommy can hold her hand and, they're sharing the same genetics. That's the problem.
Right.
Nobody's opening up. And the ones that are opening things up, they don't have the names like they should have right now.
Exactly. I mean, right now, unfortunately, with the market, it's a name sell. This cross this with this is going to sell because it's all got name recognition, and people go, I've seen somebody pull a frosty plant from that, so that's what they associate with it. Not to say that they searched 1,000 different cultivars to find that one golden standout keeper. To show off to the public. So you're correct. And, there's a lot of playing in the same sandbox, and that's, safe because it sells. And, most people that are selling seeds are out for a business. There's a goal to it. So they, stay with what's safe, and you see very little introduction, but, you're starting to see a lot of, collectors going out and collecting some land races and bringing them to market, and hopefully you'll see a lot more breeders starting to utilize those tools that are out there and have been kind of forgotten.
Yeah,
I think us growers got to help with that too, because a lot of us don't run anything past 9 weeks. Like I was one of them at one point, but I got a couple of phenos now that go 15, 16 weeks that I keep on the back burner. Things like that. I got those from Mountain Organics, but a lot of us ain't gonna have that space or be able to do that. That's one of the reasons why I'm moving, because I need that. I want some of those older things that people don't want, or that
And that's a lot of my goals. I mean, I love sativas. Landrace sativas are my thing. I love them. But not everybody, like you said, has 15, 16 weeks to have their space utilized with a cultivar. So that's a lot what I'm working with right now, is trying to shorten the time of some of these long sativa cultivars that I have, and hold true to their true nature.
My problem isn't the flowering time. My problem is the roof room in my tent indoor.
Right. Yeah,
because some of them do get about 8 foot. They get big.
Yeah, I got a chopper rare that really likes to stretch big, and I got to chop it like almost in half. I love them land races you was too.
Excellent. Alrighty.
I'm definitely a big fan of land races. I've been trying to chase back land races as far as I can go. Anybody that I know that's had any kind of land races, I chase it down. I'll go get it. I mean, if they tell me they got some old seeds from Nigeria back in the day, I want them. I'm running them. I'm hunting them.
Right.
And I've been doing it for years now. I mean, it's fun. But you do find them weirdos and them herms and all that. They're in there.
Oh yeah. Oh yeah. Just,
yeah.
Even single source seed stock that I've got that was sourced, from a single farm that's been growing it for forever. You would be surprised on how much variation there is in that stock, and that stock alone has to be worked until you can bring it out and use it in a cross.
Right, because my experience with those is you don't get the prettiest buds, but dude, those things will fuck you up like a soup sandwich. Like for
Yeah.
real.
Yeah, there's a lot has been bred out of modern cannabis.
So what would my accidental cross that I made be considered, like an F1 then, or?
Yes. You know, it doesn't matter really the generation of the plant. As if they were from separate seed stock and you brought them together, that's an F1. If they came from the same seed stock, like brother and sister seed stock, then it would be considered an F2 generation is what you would be making.
Yeah, they were totally different breeders from like opposite places of the world.
Okay, yeah, that would be an F1.
She was an accident, but goddamn, does she stink? Super chirpy.
I can probably name 10 different things that were happy accidents that are mainstream things. You know, ChemD being one of them.
So we got a question for Peter. Something about the coupon code over here in chat's not working.
Yeah, I just had to jump off there for a second.
Henry Johnson is trying to buy a unit.
All right, Henry. Drop me a DM, and I'll have a look into that. That's strange. That should be fine.
We'll have to get on that. That's about it for today, guys. I think everybody's doing well. The plant's looking good. Everybody's looking pretty and beautiful. So we'll have a good week. We'll meet back here next Sunday, yeah?
Yeah, take it
All
easy.
right, everybody, y'all have a good week.
Yeah, thank you for having me on.
Yeah, no, and if you want to carry on just shooting the shit, stay on. Don't dive off just yet.
Right on.
But for everybody else, thanks for joining the show, watching once again.
Good evening.
Cheers, chat.